Hello and welcome to another episode of TWIML Talk, the podcast where I interview interesting people doing interesting things in machine learning and artificial intelligence. I'm your host, Sam Charrington. All right, everyone, still here in Vancouver at NeurIPS, continuing our coverage of this incredible conference, and I've got the pleasure of being seated with Blaise Aguera y Arcas. Blaise is a distinguished scientist with Google AI. Blaise, welcome to the TWIML AI podcast.
Thank you so much. Thanks for having me.
Absolutely. So you are doing an invited talk here at the conference tomorrow morning on social intelligence, and we're going to dig into what exactly that means for you. But before we do, I'd love to get a bit of your background.
Sure. So it's a little motley. I started off in physics as an undergraduate at Princeton, and I studied physics and applied math there. I took a year off between my third and fourth years because I was not a very good student. and I really started to get into biophysics pretty heavily.
During your year off or after?
During.
Okay.
During, or a little bit before and then during. So I worked for a little while in Stan Leibler's lab there. He was working on bacterial chemotaxis, and that actually is going to figure a little bit into my talk tomorrow morning. So it's the behaviors of the intelligent behaviors of bacteria and how it is that they find food. They're obviously a really small, simple system, but maybe not quite as simple as people think.
Okay.
And then from there. My next advisor, Bill Bialik, is somebody with a physics background as well, but also a computational neuroscientist. And he ran this course in Woods Hole at the Marine Biological Lab called Methods in Computational Neuroscience. Methods in Computational Neuroscience. Sorry, I don't know if you're familiar or how many of your listeners are with MBL, with the Marine Biological Laboratory, but it's this place where
Princeton.
No, it's on Cape Cod.
Okay.
So it's right on the elbow of Cape Cod, across from Martha's Vineyard.
Okay.
And it's this little tiny town. It's very cute, and there's this kind of ramshackle lab that's been there since the 19th century. That a lot of visiting sort of neuroscientists and biologists have been going to for many years. A lot of really basic discoveries in neuroscience were made there.
Oh wow.
So it's kind of this cool place, and at this course at Methods in Computational Neuroscience, I met. My now wife, Adrienne Fairhall.
Oh, wow.
So she also came up in physics and studied originally chaos and turbulence and fluid dynamics and things like this, and was making the switch to computational neuroscience. So we met there. And then she ended up getting a faculty job at University of Washington, which is how we ended up moving to Seattle. And around that time, I started a company and was no longer really part of academia at that point, and the company got acquired by Microsoft a couple of years later.
And the company was doing computer vision type of work, or
somewhat.
Somewhat.
It was doing sort of multi-resolution representations of documents of various kinds. So it was a combination of wavelet-ish kind of tricks and UX. I
think wavelet is like kryptonite for me. That was the hardest thing that I studied in grad school. For whatever reason, it was very difficult to grok.
It was hard. My advisor in grad school in applied math was Ingrid Dobeshi, who was one of the inventors of wavelets.
Oh wow!
And
that might have helped.
Yeah, it helped. She was absolutely wonderful, very very smart, very kind, and I think one of the greatest living mathematicians. If I I don't know, maybe I'm biased, but anyway, Microsoft acquired it, and I did immediately turn the team toward more kind of computer vision-y things right after that. So Photosynth, which started off as the phototourism project by a University of Washington professor and a Microsoft research scientist, together with their grad student Noah Snavely, was doing 3D reconstructions of environments from 2D images. And that was really my introduction to computer vision. Okay. That was very classical. It wasn't like deep nets or anything like this. It was geometric computer vision. But I kind of fell in love with that field. And ended up at Microsoft, sort of doing a lot of leading of teams doing that kind of work. So Microsoft's OCR team and their kind of photogrammetry type teams, the teams that ended up doing a lot of work for HoloLens, for tracking the head using outward facing cameras, all that kind of stuff was part of my team at the time.
Oh, wow.
So I was at Microsoft for 7 years. I also was the CTO of Bing Maps, which also had some kind of computer vision, VR, photogrammetry kind of stuff going on.
Yeah.
And Bing Mobile. And then I went to Google. That was 6 years ago.
I've come across so many people that are in this field that have some connection to Bing.
Yeah, yeah. Bing. I mean, I shouldn't badmouth. I mean, it was creative and scrappy at the time. You know, whether Microsoft was really committed to running these things, I guess, you know, is anybody's guess.
Right.
But yeah, I mean, one of the reasons that I ended up leaving Microsoft was because about 6 years ago they had just kind of lost the phone war, and it became clear that they were going to be moving away from being a consumer-focused company, and they were going to start working on just, you know, enterprise stuff, and that wasn't that interesting to me. Okay. And that was around the same time also that the whole deep learning revolution was really getting into full swing. Mm-hmm. And I was very excited about sort of machine learning and computational neuroscience reconverging. Yeah. And Google was the obvious place, you know, where the kind of hotbed of a lot of that. So
nice. So what do you research at Google?
Well, at Google, I started a team called Cerebra, which is not a name that we've generally used in public, but that's...
Not at all heady.
well, it's the plural. Thank you. It's the plural of brain. So there was a brain team already that, you know, Jeff Dean started the brain team a few years before. And I went to Google to start a team that would take a much more decentralized approach. So rather than one brain, it would be many brains. Everybody would have a little brain. And I had a kind of very augmentation-focused point of view, you know, that rather than having, you know, one giant AI running in a data center, these things would have to shrink. They would have to democratize. They would have to go into devices, run locally. I had a lot of reasons for really wanting to push in that direction, including privacy, which I will talk about a bit tomorrow. Okay. So mobile nets and a lot of these kind of efficient ways of running neural nets locally came from our team. Oh, wow. I again am running the groups at Google that do things like OCR and face recognition and a bunch of other sort of image understanding primitives. Okay. But we also power a lot of AI or ML features or whatever you want to call them in Android, and also on other kinds of devices, including these little kind of Coral boards, which are sort of an IoT kit for doing local.
Yeah, those AI. Were, I think those were just, well, I guess it's maybe half a year ago at the TensorFlow Developer Conference.
That's right.
I think I have one.
Yeah, that's right. So yeah, we're very excited about those.
Cool. You mentioned OCR, and of all the things that we've talked about, I think of that, or it's probably easy to think of that as a solved problem, an old problem, but there's probably a lot of, I guess even just saying it, there's probably like this last mile problem where in order to get to usable or better levels of accuracy and performance, kind of those last few percentage points are really hard to get to.
So yeah, you say, I mean, it's a solved problem, and yeah, I mean, it's good enough, for practical use. There are a lot of engines that are good enough for practical use.
Yeah.
But of course, extra percentage points are always useful. So a little bit more is always better. But also, the OCR team that I ran at Microsoft was still using a lot of these classical techniques that would first, they would have a whole pipeline of different stages. First, segmenting out letters, and then doing template matching, and then using language model, all kinds of stuff like this. And the direction that I think and that the people in the OCR team believe are really the most fruitful now are much more end-to-end and much more neural. So imagine it's more like a scanner that scans the entire line, maybe bidirectionally, and emits a string of characters, kind of like a speech engine might. If you do it that way, then joined letters and ligatures don't matter. Right. Cursive doesn't matter. Handwriting and in print could be the same. Arabic and other languages that don't have good distinctions between letters. I shouldn't say good, but that don't distinguish clearly between letters and have a more cursive sort of approach. All of those things work. And that sort of generalness, and also just weird fonts. There are a lot of things that are easy for us to read that a classic OCR engine can't. Right. So thinking about it more like a real vision problem of, with the brain behind it, as opposed to just a classical kind of letter clustering problem with the language model tacked on.
So is the focus of that work today achieving the level of accuracy that we previously achieved with traditional approaches, with deep approaches, or have we
that is
met or far surpassed that?
Yeah, we've already far surpassed.
Okay.
Right. I mean, the goal now is to be able to do that in a way that is compact, real-time, runs on devices, it doesn't have to be told what language something is in or what kind of script, has a unified model for every imaginable kind of, those kind of goals, right? So the kind of things that a person can do. But yeah, the neural methods have long surpassed the classical methods.
Got it. Yeah, Jeff has been on the podcast previously and has mentioned that the transition from traditional machine translation to neural machine translation resulted, among other things, in increased performance and a reduction in the size of that code base from 500,000 lines of code to, I forget what the number was, 50 or something. It was like an astounding number.
Right.
Is there a similar...
It's exactly the same.
Exactly the same.
Exactly the same story. End-to-end learning, the code it suddenly becomes very small because all of the structure, all the statistical structure in the thing is being learned rather than coded. And it means that a lot of the assumptions that you might make in that code don't have to be made at programming time,
right?
Like letters being distinct or being read from left to right, or or not being slanted, being able to be kind of characterized in terms of connected components or boxes and so on.
Right. And so your invited talk here at NeurIPS is about OCR.
No. No, the team is pretty big, and OCR is only 3 or 4 people, I think.
Yeah. It is in fact about social intelligence,
yes.
Which is more related to the concept of cerebrus and many brains, I imagine. What exactly are you discussing in the talk?
Right. Well, it's a wide-ranging talk, and I guess it has the shape of a lot of very broad considerations at the beginning and then some specific technical work in the middle, and then maybe broadening it back out a little bit at the end. But the broad themes are that, I guess we've gotten to the point, like with OCR, that if you know what the goal is, if you're able to score or to make a loss function for what you're doing, and we have lots of training data, like we do with OCR or faces or whatever.
A couple of minor considerations.
Minor considerations, yeah. Obviously, they're not. Well, but this is the thing. I don't think that they used to be considered, all that limiting. Right. So in the days of, I don't know, the Dartmouth summer project or something, people were like, we don't know how to solve AI. Think about it. Only brains can do things like understand language and writing and so on. Surely, if we figure out how to do those kinds of things, we'll crack the secret of intelligence. And now we're like the dog that caught the car. Any of those things that can be characterized cleanly, we can achieve superhuman performance, basically. I mean, I'm making a slightly forward-looking statement. Not all of these things are superhuman yet, but it's, like if you say something isn't, then like next year it will be.
Right.
So like we've solved it, and yet none of the systems that we've built are intelligent in a way that you or I would recognize as intelligence.
Right.
It's just functions. It's just regression, really, in the end. And that's why we say, like AI or ML/data science. And in a way, the projects of AI and data science could not be more different. Data science is just about modeling data, right? And AI is about making minds. I mean, on the face of it, like, really, are these the same thing? So, I think if there's a single theme for my talk, it's like, well, what is that gap, and why? And why is it there? The reason that I called it social AI or social intelligence was that I've come to believe in the last few years that that sociality, which is to say our interactions with each other, are not incidental to intelligence, but are actually fundamental to it. In the sense that life isn't a one-player game. It's not like we evolved intelligence as an adaptation in order to get by in a really hard video game environment where nature is trying to kill us and we have to outwit it. On the contrary, like an individual human is a lot worse at outwitting nature than our ape ancestors. Like drop one of us naked in the jungle and like only a handful of us will make it. Maybe the piraha, in Brazil or something. But there are very few people who can make it.
We get to watch it on TV.
Yeah, exactly. So our environment is each other, and there is a famous researcher, Robin Dunbar, who proposed the social cortex hypothesis way back in the 90s. And he's not the first. Frans van der Waal and Frans de Waals and many other researchers have had the same kind of idea that we are, essentially the role of intelligence is to model others. And since you and I are of the same species, it's useful for me to be able to model you and to understand what you're going to do. And so my brain will grow bigger in order to model yours, but we share genes. So in the process, your brain grows bigger as well. And you have a kind of, arms race or a feedback loop. And that's how you get these kind of intelligence explosions that we've seen in the apes and in cetaceans and dolphins and whales and some other species.
Echoes in a lot of ways Yuval Harari's from Sapiens idea that our kind of core achievement as a species is the ability to collaborate and communicate goals and stories to one another and kind of project forward. Yeah,
I absolutely agree with that point of view. There are a bunch of recent books. Harari's are among them, but also Nicholas Christakis, Blueprint, The Human Swarm, Moffat's book, Pat Churchland's book, Conscience, a number of books that I think really, sort of start to expound that point of view. But I don't think a lot of that has been heard in our community, in the kind of AI or ML communities.
So you're proposing this idea that intelligence is a collaborative idea. How does that manifest itself? Or what are some of the kind of next layer points that you're making in the talk?
Sure. Well, so one of them is that when you kind of zoom out to think about not just the intelligence of an individual, but the intelligence of a group, of a society, which, most of our achievements are, of course.
Right.
Then you are in a multi-agent kind of universe, if you want to think about it in kind of our terms. And in a multi-agent universe, even if every individual agent is doing optimization, in other words, has a clear loss function or objective and is doing, gradient descent to optimize it, when you zoom back and look at the whole, that is no longer the case. And you can see that even in a really simple example, like, the most minimal ecology you can make is an ecology of two things. And GANs are an example of an ecology of two things, right? You have an actor or an artist and a critic, and their goals are different and they're kind of misaligned, right? The artist is trying to fool the critic, the critic is trying to winkle out, to suss out the artist. And when you look at what happens in the interaction between those two, they pursue each other and there's a spiral, so this is a dynamical system. And dynamical systems have chaos, they have vorticity, they have limit cycles. And that does not look like gradient descent anymore. Like if you look at the dynamics of gradient descent, it looks like a curl-free field. And
curl-free?
Yes, meaning that there's no twist in trajectories. Everything goes downhill, so trajectories have no curve in them. Whereas these GANs, that's why they're so quote unquote hard to train, because they have predator-prey kinds of dynamics essentially. So what happens is a lot more complicated.
Right.
It so happens that GANs were invented to have a fixed point that coincides with the optimum of a function that can be written down, which reproduces a distribution P of X. But that's just an artifact of how we cooked that particular one up. And in fact, it's not as if GANs, when they converge, are in general necessarily at such a global optimum either. So that's like the minimal ecology. And when you consider that we're made out of cells, neurons are cells, they each have their own objectives and so on. It's not just ecologies when you look at, say, multiple people that have that property, but a single brain or a single thing is in turn composed of sub-things that have their own goals and agendas. So it's turtles all the way down and all the way up. And when you start looking at it that way, then I guess I'm not saying that optimization is dumb or is a bad way to look at things, but it's more like that's just a local force. And when you look at the system, the behavior of that system cannot be determined by just looking at those local forces. You have to look at the entire picture.
I mean, it's especially fascinating when you think of it in the context of research like, how much of our behavior is controlled by our microbiome.
That's right.
I was just watching some TV show, maybe it was like a Earth type of show that showed, how some parasite would infect these ants and cause them to do these zombie-like behaviors, mind control.
Yeah, that's right.
Which I think underscores your point that, so much, behavior, manifest behavior that we see is controlled by kind of the interactions of things as opposed to, some optimization function.
Exactly. And the boundary between parasitism or predation or exploitation and symbiosis is, not only is it a fine line, I'm not even sure the line exists.
Well, certainly not if you step back and look at a system.
Right.
What is the system's optimization function may be very different from that of the ant.
That's right. But even if you look at things that, even if you try to make firm boundaries around, say, an individual person, I mean, like there was a recent discovery that I thought was really cool about the ARC virus. So there are a lot of retroviruses that have been incorporated into our germline, into our genome, and that are passed on. Some of them for many, many hundreds of millions of years, and the ARC virus is one of those. We've known that it's there for a long time, but nobody knew what it was doing. It was finally caught.
Just so I understand this, when you say passed along or kind of incorporated into our gene pool, meaning the body fundamentally creates this virus as part of existing.
That's exactly right. Like the virus is no longer distinct from your genetics. It's not. It's not that it's endemic in a population and gets passed on. It's literally part of your DNA.
Okay.
So these are retroviruses that inject themselves into the DNA and into the germline and propagate along with you, just like mitochondria. Those are bacteria, of course, right? But we're full of these kind of symbiotic things. And ARC, it turns out, it was caught in this electron microscope actually forming its viral coat and forming its capsid.
Okay.
And if you knock it out in mice, they can't remember anything longer than 24 hours.
Wow.
So like they're involved in some way that we don't fully understand in memory formation.
Huh. Wow.
Some people think that AIDS, it was kind of on the way. I mean, hopefully we've now sort of controlled it, but we might have essentially been witnessing a kind of event that has happened many, many times in human history, wherein a virus begins very virulent and devastating, but eventually kind of goes global, as it were, and becomes incorporated into our genes for good.
And so to make this more concrete, perhaps.
Yes.
Well, maybe before, a pit stop before we go into more concrete and technical, it calls to mind, obviously, I guess, kind of a lot of the research in, I think, maybe the 80s or 70s, like swarming behaviors and things like that.
Yeah, that's right.
Which I'm imagining are very simplistic models relative to the things that you're thinking about.
Yes, and
also
no.
Tractable.
Yeah, that's right. So that's if I have time tomorrow, I'm going to show two sort of technical things. One of which is very simple and is very much along those kind of 80s swarming sorts of lines. It's a simulation of bacteria. So trivial system, the learning is very, very simple, and so you can characterize it completely. And the other one is about neural nets and update rules for cells and synapses. So that one applies to to the kind of systems that we build. So the bacteria one, it's very much inspired by that kind of Santa Fe Institute, sort of work from the 80s, 90s. And the idea there is, bacteria have E. coli in particular, can either run or tumble. So when they run, they're going in a straight line, and when they tumble, they randomize their direction. It has to do with the direction the flagella are rotating. And they're too small to have a sense of like the global environment. They can just go straight or kind of reverse, kind of like those RC cars that have a like go straight or reverse and turn kind of mode. So it's a 1-bit output. And so I did a little simulation of them, and they have a food source that moves around, and if they don't get enough food, they die. And if their energy level gets high enough, they reproduce. They can also conjugate when they touch. They can swap a little bit of DNA, and they undergo some mutations. So it's artificial evolution. The first experiment involves thinking about the genome as just being the Q table. In other words, in RL language, right, just the table of stimuli to behavior. And you can see how with this kind of evolutionary pressure, they evolve to follow the food. There's a kind of there are algorithms that let you do that. Essentially, they involve tumbling more when you're in the food and running more when you're far away from the food. Okay. And statistically, that will make the colony of bacteria follow the food around.
I'm imagining the picture that comes to mind for me is the kind of the classic picture of an RL agent. I forget the game, but it's like a boat game where the RL agent kind of learns that it can rack up points by just spinning around in this particular place.
Yeah, that's right. Well, I mean, one of the fun things, of course, about RL is that, and any kind of machine learning really is that it'll if there's a way to cheat, it'll figure it out.
Right.
Like life.
Right.
That's right. So it figures out how to follow the food around this way. Things get a little more interesting if you add an additional output and you let them emit an extra chemical that they can also sense.
Okay.
So that's done by bacteria. They have chemosignaling. So in the rules of the game that I set up, if they're signaling, they're losing health faster, so it's costly. It's costly to signal. So you'd first think that, well, the first thing they'll learn is to not signal because it has no advantage for them, basically, and it burns energy faster. But they don't. They keep signaling. So in generation after generation, restart after restart, they almost all of them retain the signaling capability. And what you realize, of course, is that they become a superorganism when they start to share genes. And so thinking about them as individuals or as a single organism, as a tissue, it's kind of like in the eye of the beholder. There's no right. That's not determined by the rules of the game.
And so, are we talking about the observed behavior in the game,
in the simulation,
or in the
in
thing
real
that you modeled? Okay, yes and yes.
Yeah, yes and yes. In the model, you can reduce everything to very, very simple principles and see all of those behaviors emerge, which is fun because then then it becomes clear that we're understanding something. So yeah, they signal to each other. They obviously are behaving like a superorganism. You can't say what is the agent. Is it one? Is it many? And it's also kind of hard to say what they're optimizing. If you take a very Darwinian red of tooth and claw kind of perspective, they're like, well, they're maximizing their energy intake. Well, but who is maximizing their energy intake? Is it an individual bacterium? Is it the whole colony? How do you think about, the population or the size relative to what is being taken in? What does optimization actually look like?
Because in simulation, you observe behaviors that are suboptimal either individually or globally, but optimal otherwise, or?
Well, even talking about optimal is hard. So,
according to the optimization that you propose, that it's all about energy intake and thus kind of speed of reproduction.
Well, all I said was that they die if they go to 0 and they reproduce if they get to 1. I didn't say what was being optimized.
Okay.
So, the thing with evolution is that what you see is what makes it. What persists exists. We're not actually writing down a loss function. So you can ask what is optimized, and that's actually an inverse reinforcement learning problem. In other words, you have a Q table, now you back out.
What's the policy?
What's the reward? And I kind of cheated in order to get, so IRL, inverse reinforcement learning, is actually a hard problem in general, but I cheated by switching things up so that rather than the genome being the Q table, now the genome is the reward map. So they essentially evolve a reward system or an emotional system, if you want to think about it that way, and we see well, what's the reward system that actually results in in survival? And what comes out is kind of what you would expect, which is that in general, you look at this many, many times, death is bad, signaling is bad because that's that hurts. You lose health,
energy,
right? Mating is good, survival is good, food is good. Those are positive, these are negative. So it's kind of what you would expect. But the surprise is the error bars. So when you look at specific winning solutions, that is solutions that converge and that persist, they're all over the map. So those are averages that I just gave you, but the variance is huge. There are all these different reward maps that all work. Some of them result in...
Meaning dying off is a lot harder than continuing, in a sense?
Well, the set of...
If you can define harder.
The set of emotional systems or rewards that work is small relative to the total imaginable space of reward maps. But it's also very varied.
Right. And there's not just one.
There's not just one.
Right. Okay.
But there are many. And some of them involve lots of exploration. And therefore, death is not bad for those. They're not afraid of death in some sense. Some of them are very conservative and therefore have smaller populations. They hate death. They don't signal very much. They follow the food around really closely. And all of those are viable. So, even
so this is kind of one data point, just so we don't get stuck in time on this thing, because I could ask you a bunch of questions about this particular thing. This is one data point that informs a larger perspective. What's the other? You mentioned a second example?
Yes. So, all of this is really just a kind of extended example of, of some of the problems that arise when you try and look at a real evolutionary system in terms of optimization and try and ask basic questions like, what is it optimizing? So I then take that to the home, I guess, to the thing that concerns all of us at this conference, which is, all right, so how do you train a model? There's been a bunch of work in recent years on meta-learning, which involves learning to learn. So learning maybe what the update rule is at a synapse, or learning how, not assuming that the learning algorithm is fixed. I really like that approach because, our genome basically has in it the learning rule for our neurons and synapses, and that evolved. So I began playing around with systems that evolve, that use evolution to determine the rules for neuron state and synapse state over time, and therefore that have to learn to learn. They learn to learn on an evolutionary timescale, and they learn on a behavioral timescale, if that makes sense. So the learning to learn includes instinct and imprinting and other kinds of things, and the learning to learn, establishes the ability to go from stimuli to generalization. And if you do that, you basically have a little kind of LSTM at every neuron and every synapse that has local state and has its own timescales and so on that it learns evolutionarily. Then you can get very, very fast learning of standard kind of machine learning models. I give some MNIST type examples. So learning from very, very few examples. And that's interesting if you just think about it in terms of optimization. This is in the spirit of some other work that has been done. Ravi and La Rochelle wrote a really nice paper in 2016 or 2017 that did something similar. My version of this is a little more general in that it's not designed necessarily to optimize. It's kind of broader. It's just like any old, it's a very general kind of synapse rule and a very general cellular evolution rule. And I don't
so I understand this, you've got the picture that's forming in my head is kind of multiple agents or entities that have some kind of embedded, memory sequence, LSTM based thing. And in their interactions, they can essentially learn and solve MNIST type problems.
Well, I do show an example like that, but an even
that's
simpler.
Not at all what you're saying.
Well, the simpler version is as follows.
Okay.
Imagine that you just have, in one neural net, a single kind of LSTM, a single set of weights for an LSTM that live at every cell and every synapse. It's like your genome, right? So that determines how the cell responds to inputs and how the weights change.
So it's kind of a dynamic weight model based on the parameters of the LSTMs at each of the neurons, essentially.
That's right. And it's a common LSTM, so it's a small. It's like the difference between the genome and the connectome. Genome is very small, connectome is very large. The LSTM parameters are the same everywhere, like a convolutional net, kind of.
Okay.
And so they comprise the genome of the neural net, and whatever weights it learns over time are the connectome.
The same everywhere in structure, not in value.
That's right. That's right. So different state at every neuron and synapse, but the same weights.
Right.
Right on the LSTM.
Right.
Which in turn determine the rule for updating the weights in the neural net. A little confusing, I know, but
it's hard to do without the whiteboard.
It's a little hard without the whiteboard.
We run into that a lot.
I'm sure. So yeah, how do you train? How do you determine the genome? Well, now you do have to have a population. So you have a population of neural nets that in the beginning have random genomes, and they attempt to learn, meaning you feed them MNIST digits, you feed them an error signal, and the ones that do a better job survive and reproduce, meaning they share their LSTM weights and yield a new generation, and the ones that do poorly die off. So you can use evolution strategies to do this. I use CMAES, or you can use more classical kinds of evolution.
What's CMAES?
CMAES is so the ES is evolution strategies.
Okay.
And CMA is a particular variant of this that assumes that the set of parameters is a Gaussian blob. Okay. And essentially estimates the gradient of the fitness along that Gaussian and uses that to kind of make a new Gaussian in the next generation.
Okay.
So it's a toy model of evolution, and it's a very robust optimizer for moderate dimensionality. So you do that with the genes, and you end up evolving a kind of net that learns really fast, if that's what you make your fitness function. Like learn from the minimum number of examples, learn as fast as possible. So like in a normal MNIST kind of setup, you measure how much training data in terms of how many passes over all 50,000 training examples or something. This one, you measure its performance in terms of how many digits you show it. So like after you've shown it 10 or 20 digits, it's already doing pretty well. So it's really, really, really small n.
And are you able to introspect into it and understand what
the hell it's doing.
Learning? What the hell? Yeah, exactly.
Yeah. Well, I haven't done enough of that yet. But
I'm envisioning like the texture and feature maps of a neural network.
That's right. Well, I don't think that what it's learning, what the network is learning, that the weights are weights is an ambiguous term here. I don't think that what the synapses get set to is particularly different from a normal neural net.
Okay.
What's different is the update rules.
Yeah.
So and the update rules are governed by a little LSTM. So figuring out what that is doing is, is kind of like a biology problem. It's actually not that, it's not that trivial. But you can do things like, normally when we do backprop, we assume symmetric weights, meaning, that you use the same weights when you're back propagating as when you forward propagate. You have to, otherwise you can't take the derivative. But we know that in real brains, that's not how it works. Like, signals don't propagate back through synapses in order to update neurons.
Right. And so usually we think of MNIST as this toy problem, but for someone that works on OCR, it's particularly relevant.
Arguably.
Like, are these things that you can you have a path to actually putting in production, and for what?
Well, in this case, the goals are to be able to learn from small data, which I care about from the point of view of privacy, and I care about because, I think one of the Achilles heels of deep learning as we do it today is its reliance on massive amounts of data. And the reason we have that reliance is because we don't have very sophisticated learning rules, and they don't embed any priors that have been learned over evolutionary time the way ours have about the world. Right? It's all of those priors and all of that intelligence that lives in our genome that lets us learn so fast. So, this isn't an attempt to say like, well, can we reproduce what evolution did in order to learn statistical priors and learning rules that radically outperform the feature-engineered rules that we have today, ADAM and AdaGrad and SGD and all that. So that's interesting, just from the point of view of
I get why
normal.
We want it. My question is there a catch? Right? Do we just throw LSTMs in everything, and now everything converges way more quickly?
Yeah, why not? It's much more computationally expensive to do that, of course. But on the other hand, if you can learn from many, many fewer examples, then it's still a good thing,
right?
Even computationally, and certainly from the point of view of data. Is there a trade-off? Yeah, absolutely. The trade-off is that when you're learning from very few examples, that means that you're bringing much heavier weight and sometimes rather opaque priors to bear on the problem. So you're subject to more cognitive fallacies and all kinds of all the things that humans are subject to.
So all the issues that we talk about as bias that's being introduced in our data distributions are potentially magnified manyfold because we're training on much less data.
Possibly. Although I guess I would say a couple of things. One is that when you can learn from very small amounts of data, then you can perhaps be a bit more careful about how you curate that. But and also, of course, the fact that the genome is very small means that you maybe have a little bit more hope of understanding how, what those biases and meta priors are. So I think it's still positive from the point of view of problems like ML fairness and so on. But that's definitely something that one has to look at very, very closely. Because newborn babies will react with fear to snake-like objects. So that even at a very high level object recognition kind of level, there's something in the genome that that makes snake-like things scary. And so you can imagine the problems that can arise from having priors like that, hidden priors like that, in the genome for learning nets. So yes, that's definitely an issue. But at a more meta level also, to connect this to the bacteria stuff, what you can imagine is that this rule, it's not just a learning rule, it's actually a rule for how brains or how behavior works. And in that sense, it's like an emotional system. What has been learned by evolution is what is good or bad as well, locally. In that sense, I think that this is a route toward real AI in ways that I don't think we can do with, I don't think we can get to that by handwriting either update rules or fitness functions or loss functions.
And so in the model we were just talking about, the individual agents, actors, whatever you want to call organisms, are these LSTMs.
Right. So that's kind of looking at a neural net as a society of neurons, if you want to think about it that way. You can also then take these brains and put them together and have them teach each other, the kind of things that you were pointing out, right? Have them interact socially. And that's also super interesting. And I think that that's how you actually get emotional systems that work. So I have come to the belief that our route to, I don't know what to call AGI, I guess, right? But our route to real brains has to go through this
social intelligence.
Yeah, it has to go through the social route and stopping hand defining the loss functions and the update rules.
Interesting. One of the things that is common between the swarm intelligence approach we were referring to earlier and what you just described is kind of everything's the same. Like you're building these systems out of components that are the same and groups of components that are the same. Whereas, maybe the counterexample that we've talked about is GANs, where you've got these two distinct things with different goals.
Yes.
Have you started looking at more heterogeneous systems?
Yeah, I have. So even within a single neural net, I've often played with having different genes for every layer, for example, or having different brain areas that have different genes in them that are interacting, with that are sort of feeding into each other with asymmetric weights that are whose interactions are learned, or having multiple species together that have entirely different kinds of inputs and outputs, and that so it gets into a very a very artificial life kind of paradigm. So yeah, I think those are all really interesting routes. I mean, the challenge with a lot of this is, of course, that we've really relied on the fact that these things look like scores or games for a long time in order to talk about like what is state of the art, what is better or worse. It's research groups competing or collaborating with each other, right, with a well-defined metric, and it's really hard to take these more social and organic kind of approaches and come up with the same sort of clear metrics, for what constitutes progress. So that's one of the big challenges that I want to kind of leave the audience with. How do we keep that sense of clarity of progress about how we're advancing in our understanding and our ability to solve these problems when we, I believe inherently need to start looking at things that don't have such well-defined scores.
And is that because the things that we should care about are internal metrics of these social entities, or if we want to get kind of near-term-ish value out of them, we're applying them to problems that have some type of score associated with them?
Yeah.
Like, the things that we've been doing, like translation and OCR and, playing games and things like that. Why aren't those metrics sufficient for these types of systems?
Yeah. So it's both of the things you just said are correct. I mean, I'm not saying that metrics are a bad thing for OCR, or for face recognition, where it's very clear what is good or bad. So the problem of ML fairness for face recognition, for example, is now much discussed, and it's actually really well posed. Mm-hmm. Like if it doesn't work as well at distinguishing faces for some group of people that we define socially, then that's a problem. And the answer is clear. Like sample better in that space, measure better in that space, and and set a higher bar for for everything to perform equally well. Mm-hmm. The problem is that most real stuff doesn't actually fit into that paradigm. What is the loss function for for a credit score, for the correct assignment of a credit score, or for couples counseling, or for good or bad art, or for how to rank notifications on Android, for that matter. So like really bread and butter stuff. You know what I'm saying? Like if you just optimize for engagement.
I can tell you a thing or two about the loss function for ranking notifications on Android.
Well,
and actually they're rules.
Yeah. So at the moment, it's unfortunately a lot of hard rules that themselves. I mean, we're back to Q tables again. Like if you try and back out using inverse reinforcement learning,
right?
Like okay, those rules are all kind of carefully considered. Like what are they optimizing? You cannot write down what they're optimizing for, right? They're coming from a whole bunch of different socially constructed intuitions. So
kind of your point is you're proposing this potentially much more powerful paradigm, and with this much more powerful paradigm, we should be able to push into areas that we can't apply.
That's right.
I always falter saying traditional machine learning since we're
right, but machine learning as we know it today.
Machine learning as we know it today.
That's right.
Which is very much based on kind of being able to write down these loss functions.
That's right.
But, we're kind of it's a little bit of a chicken and an egg. Like we can't write down the loss function, so we don't know how to apply these new things or even how to measure success with these new things.
Well, and what I would argue is that, it's social, right? So the point of ranking, I mean, if you were doing the ranking, and you were like a little person living in somebody's phone doing the ranking for them, and you were able to consult also with the other rankers, and everybody else's phone using federated learning or something like that, then the fundamental tool that you'd want to be able to do that effectively is empathy. You know, it's not like maximize engagement, you know, or some kind of stupid, you know, quantitative measure like that.
Right.
It's more like, you know, be good for the humans.
Right.
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 So that requires empathy, and that requires modeling the other and using that as a guide for how to behave collectively and individually. And there may be conflicts in that, like, you know, whether you model them in, you know, at the individual level or the collective level. You know, just like in medical ethics, you have conflicts about, say, you know, I don't know, giving people antibiotics. Right, in the aggregate, it's not necessarily a good thing. If you're only optimizing for the individual, you'll do it more, and so on. The same issues come up here. So, you know, you've got to kind of create an entity that has clear allegiances and clear kind of you know, empathic goals that don't quite look like loss functions, but look more like being able to relate socially to the relevant kinds of entities.
And you're coming at this from a biological perspective. Yeah. I imagine that there are connections to many other, you know, economics and sociology and other things if you were to really get into the social aspect of this.
That's right. And dynamical systems theory and various other fields. Right. So a lot of other fields come into it.
Yeah.
Exactly.
Yeah. Awesome. Well, Blaise, thanks so much for taking the time to, you know, share what you're doing and give us a preview of what you're speaking about tomorrow. Of course, folks that are listening to this can actually go and check out the recording of your talk.
Absolutely, my pleasure. If they've listened to all of this, they've gotten a longer version that'll be the talk tomorrow morning. So thank you for asking such great questions.
Nice. Thank you so much. All right, everyone, that's our show for today. For more information on today's guest, visit twimlai.com/shows. Thanks so much for listening, and catch you next time.